This is a program on biofuels, and bioethanol is one of the important component, and in that too saccharification is one of the important step in the bioethanol production, where we concentrate on the conversion of lignocellulosic biomass into reducing sugars, and that ultimately decides the economy of the overall process, that how much of the sugars we are able to get after the saccharification process. And then finally, if it is the yield is good, saccharification efficiency is good, then we'll be able to get good amount of ethanol converted by the Saccharomyces. So to take on this hands-on session, we have Professor Vikas Beniwal, our esteemed head of the Department of Microbiology, and he has got research interests of industrial microbiology, enzyme technology, and bioremediation. And he is serving as a professor at our department since 2022. Prior to that, he served at different position in Maulana Ambala, uh, and before that also he has got vast teaching experience, starting from 2003 until now. You can call around 22 23 years of teaching experience. And if you talk about his educations, his BSc from Rajasthan Agriculture University Bikaner, and MSc from GJU Hisar, and PhD again from GJU Hisar. And if you look at the citation profile, he has got more than 100 publications, including research papers, book chapters, one book, two patents, and he has completed one research grant also. And overall citations is more than 2000, with an H index of 22. And he has guided 7 PhD students, and one is or two students, they are undergoing PhD as of now. So I welcome Dr. Vikas Beniwal for this practical session in this GAN program, and really thank to him for sparing his valuable time. So I request Dr. Vikas to take the session. Thank you. Uh, so thank you, Dr. Professor in absentia, for the introduction, and good morning to all of you. So today we will be going for the practical session of cellulase activity, cellulase enzyme activity. And before going to the practical session, just go to the basics. We want to go through the basics, that how we can estimate the activity. So we will be going through that, and then we will be following up in the lab with this practical session. So the topic is estimation of cellulase activity. So before going to the cellulase activity, as Professor Surinder Singh was saying, that main motto is to convert the lignocellulose biomass into bioethanol. The first step is saccharification of lignocellulose biomass. So before acting the enzyme, let's understand the plant cell, as you all have gone through that plant cell wall. If we talk about, or we talk about the plant cell, so this is the structure of overall, you can say, plant cells. And if we talk about the plant cells, they are combined together. We call it as middle lamella. Mostly it's contain of pectin and other oligosaccharides. Then basically comes the primary cell wall. Then comes the secondary cell wall. And we all know that the primary cell wall is made up of microfibrils, and this microfibril contains the cellulose basically. And along with the secondary cell wall, that also contains
cellulose and hemicellulose, lignin and xylene along with this I can say cellulose. So all these constituent of polysaccharides are there, and we are interested to convert these polysaccharide into monomer, or more specifically we call it as reducing sugar. So our main focus is towards the cellulose, to convert the cellulose into monomer in terms of glucose, I can say, and then we can convert the glucose into ethanol by saccharomyces cerevisiae. So this is an overall representation when we can say that overall cellulose composition in the cell wall is around 30 to 60%. It varies from sources to sources, and next one is hemicellulose. It contains around 15 to 35%, followed by lignin and then pectin. So these are the constituent, and we will be following the cellulose part in this. So cellulose degradation, if we talk about, we have three basic enzyme. If we talk about, we have exogluconase, we have endogluconase, and we have beta glucosidase. So there are three enzymes we will be talking about if we are talking about cellulose activity. And the first one, if we talk about the structure of cellulose, cellulose structure is a polysaccharide, we all know, made up of a repeating unit of glucose, we all know that, and the joining link is beta 1,4 glycosidic linkage. So our main purpose is to break this beta 1,4 glycosidic linkage so that we can free the glucose, and then we can use that glucose to convert into ethanol. So to break them, the first, as I was talking about, the first one is glucanase. We call it as endo and exo basically. So exo act on the terminal basically. So we have a reducing terminal. If we talk about, we have the non-reducing terminal. It act on both the terminal, and as a result, it give us cell bios. Cell bios is again a oligosaccharide. More specifically, I call it as a disaccharide. So we are getting disaccharide linked by beta 1,4 glycosidic linkage. And if we talk about endoglucanase, so it's basically, as the name suggests, it's acting in intermediate basically, and it's breaking the chain in between. So when the endoglucanase is acting, we are getting various oligosaccharides, and our main purpose is then further to break down the oligosaccharides into monosaccharide or into reducing sugar basically. So both endo and exo, they are giving us disaccharide and oligosaccharide, and then comes the activity of beta glucosidase, and then when beta glucosidase comes into the picture, then it breaks all the oligosaccharides and disaccharide, and we are getting the individual sugar that we are interested in. And finally, when we have the monomer or reducing sugar, or in simple you can call it as glucose, now it is available to be converted into ethanol. So this is an overall representation that how we can go for cellulose degradation. Then as the topic was to estimate the cellulase activity, so we are going towards the protocol, and you will be doing, most of the students, I think some of you have the PhD title also related to this topic, so you are doing routine work. Some of you have not gone through, so you will be going in the practical session. Just I am giving a hint that how you can estimate the cellulase activity. Initially I am talking about endoglucanase activity, cellulase activity in other terms we talk it about. So we are using CMC as a substrate in that case. CMC stand for carboxymethyl cellulase. As we all know that cellulose is insoluble, so it contains a carboxy group, and that carboxy group makes it soluble. And unless and until a biomolecule is not soluble, we can't convert it into a ethanol, or we can't act upon more specifically with the enzyme other than if we talk about lipases. So carboxymethyl cellulase is there. So we basically take the filtrate and we incubate the filtrate. If I am talking about the culture filtrate, so we incubate the filtrate with the CMC cellulose 1%, that is the substrate. and then further we incubate for 30 minutes at 50 degrees centigrade. After incubation, our main purpose is to find out how much sugar is liberated, how much sugar is released. In other terms, we want to know the reducing sugar. So in that case, we use dinitrosalicylic acid, as it is called 2,3,5-dinitrosalicylic acid. It converts into 2-amino 3-nitrosalicylic acid, or salicylicate, I can say. So this is the conversion of you can say uh, this dinitrosalicylic acid that facilitates the identification of you can say the sugars. So once the densa is added, we boil for 15 minutes, then we make up the volume to 20 ml, and at final stage we go for the OD at 540 nanometer, and more the reading is, more the reducing sugar is. So basically it tells about how much sugar is released, and this is more specifically done in case of I can say endoglucanase activity. Another one is we are interested in the second activity, that is beta glucosidase activity. So as I have told you earlier that you have endo and exo glucanase activity. So you are more interested that once you have oligosaccharides in your hand, then you want to convert them into monosaccharides. So for monosaccharide, the linkage is targeted, and we call it as beta 1,4-glycosidic linkage, because cellulose is made up of 1,4-glycosidic linkage, and even the degraded you can say oligosaccharide, they all contain beta 1,4-glycosidic linkage. So to see the activity of beta 1,4-glycosidic linkage, we go for again glycosidase, beta 1,4-glycosidase activity basically, and our target is the linkage. So in that case, we take the substrate, that is para-nitrophenyl beta D-glucopyranoside, and as the name suggests, a phenyl group is attached as a beta 1,4-glycosidic linkage. So rather than a sugar, a second member is a phenyl group. So once there is a beta 1,4-glycosidic linkage, the phenyl group will be released and will be used for the production of color, that is used further for estimation of enzyme activity. So now if we start the procedure, it start with 0.5 ml culture filtrate, and then again we are adding equal amount of the substrate, it is again 0.5 with 5 millimolar, and then we are incubating it at 30 minutes again for 50 degrees centigrade, and then finally we stop the reaction by adding 1 ml of 0.4 glycine buffer with pH 10.8. This buffer works in two ways. One is it's inhibiting the activity of beta glycosidase. Another one is it helps in formation of the color. The color is due to the release of phenyl. So the phenyl is released, and at alkaline pH it is giving you a color. That the color is now measured at 430 nanometer. So more, again I say, more the color it is, more will be the activity. So these are the two enzymes we will be talking about today. We are not going towards the exoglucanase activity. So only endo and this beta glucosidase, we are talking about this in a lab. So hopefully it is clear to all of you, and we will be heading towards the practical session for this, right? All right. So we will be heading towards the lab for this, right?